What you're looking at here is the cell membrane. This is the surface of the cell membrane. All those little balls are the heads of the phospholipids. Originally, scientists thought that cell membranes were static, solid structures, but what we realize now is that they're very fluid. They're very dynamic. There's constant movement going on. So what looks like a ball pit from McDonald's there, those are all heads of phospholipids that are constantly jostling around and moving around. Now the heads are hydrophilic. They like to get near water. Beneath the heads are the fatty acid tails that are hydrophobic. And those tails, especially in this image right there, you can see the tails face another set of tails, which then have a hydrophilic head that actually faces the inside of the cell, creating the phospholipid bilayer. So cell membranes are incredibly complex structures, not this static, flat, thin little layer we used to think they were in the early days of understanding the cell. So what we're going to explore here in our next chapter, chapter 5, are cell membranes. How they work, their basic components, the function of the membrane, the arrangement of the structures in the membranes, and what happens when you stress a cell out. How does the membrane respond? Does it stretch? Does it relax? All these actin filaments we're looking at here are part of the cytoskeleton, what we talked about back in chapter 4. Those all play a role in the structure and function of the cell membrane. So let's take a look at the membrane in a little more detail and figure out the basic features behind the cell membrane. Okay, so membranes part 1. This is chapter 5 in our book. So the membrane was the fluid mosaic model of the membrane was coined or kind of determined by Singer and Nicholson back in 1972. What they figured out is that the proteins associated with the cell membrane float on the membrane or actually are embedded within or actually cross through the entire membrane, as in the case of this structure to the right-hand side of the picture called the transmembrane protein. But all cell membranes have 4 basic components. Doesn't matter what type of cell you are, bacterial, archaea, protista, fungi, plantae, or animalia, 4 basic components. So the components include a phospholipid bilayer. So the phospholipids, 2 layers of those phospholipids. They will have transmembrane proteins. These are those proteins that go from the inside to the outside of the membrane. They allow substances to move through the membrane, to either go from inside the cell out or outside the cell in. They will also have, membranes will also have an interior protein network. So this goes back to the cytoskeleton of the cell. That interior protein network provides structural support for the membrane. That's the cytoskeleton we've talked about, the different filaments associated with that. And then the 4th key feature or component of all cell membranes include cell surface markers. Now these markers act as cellular identification. They allow the cell to attach to another cell. They also allow the body to recognize the cell as belonging there. So we'll talk about things like tissue rejection. When somebody gets a skin graft, if it's not their skin, those surface marker proteins, these cell identification proteins, don't match up to their body, so then the immune system attacks it. So cell surface markers are an incredibly important structure to allow cells to work within a multicellular organism. So we'll explore these different features as we go through the cell membranes. So starting with the first feature, phospholipids. This is our basic phospholipid. We've seen them before, we've talked about them, they're back again. Phospholipid is made up of a polar hydrophilic head. Now, hydrophilic means it likes water. So that little ball structure there, that likes to get near water. It's water loving. That's what philic is loving or likes. The phospholipid also is made up of two fatty acid hydrophobic tails. Now, from the food chapter, when we talked about the foods, we talked about lipids, and they don't like water. Fats hate water. They're afraid of water. They repel water. So those two tails that dangle down, they don't want to go near water. So it's a conflicted molecule. Heads near water, they like it. Tails away from water, they don't like it. But what this does is it sets up the membrane to be a bilayer, and a fluid, movable, mobile bilayer that is constantly jostling around. That's why we saw that one image, and the image focused on the hydrophilic heads, and they were moving around because they're constantly adjusting position, trying to keep the tails away from the water. The membrane also has those different proteins we talked about. Here's some great examples. We have the peripheral proteins, we have the integral proteins, we have surface marker proteins, we have cytoskeletal proteins. Proteins are also a key feature of these cell membranes. It's not just phospholipids. Phospholipids make up the bulk of the membrane. They're more numerous than the proteins, but those proteins play essential roles. So the membrane is fluid. The fluidity of the membrane can actually change as the environment changes. If it heats up, becomes more concentrated with solute or less concentrated, that fluid nature of the membrane can actually change. Okay, so our basic structure of the phospholipid bilayer, polar hydrophilic heads always facing water, water on the outside of the cell, water on the inside of the cell, and then those tails pointing towards each other, trying to stay away from the region of water. Now, sometimes substances will force their way through those hydrophilic heads. They get into the tails, they push their way down to the other side of the membrane, but the membrane will hopefully seal itself around that substance, preventing water from getting in. So, simple example, if you guys have a bowl of Jell-O or pudding, stick your finger in it. And as you push your finger in, the pudding separates, but your finger slides in. When you pull your finger out, the pudding collapses back around the hole, seals it up. That's what phospholipids will do when anything gets pushed through the membrane. Right, now here we have a table that'll hopefully help make life easy for you guys. Table 5.1 talks about the different components of the cell membrane. So definitely make sure you remember phospholipid bilayer and the phospholipid molecules, what their function is. I want you guys to look at the composition, the function, and then examples of it. Don't get too worried about how it works. General concepts are good, but definitely make sure you know the different types of structures associated with the cell membrane. So again, this table is kind of the shortcut table. It's a good reference table, be an easy one to set up as a study guide. Can you fill in the blanks? Can you remember what structure does what type of information here? So table 5.1 right out of your textbook. Okay, so here we are looking at the proteins. Please remember, protein structure equals protein function. This is a concept we talked about at the beginning. We talked about structure equals function. We talked about it when we looked at foods, and we talked about proteins. Shape or structure determines function. If you change that shape, change that structure, you change the function of the protein. And that protein may not work, which then can have significant consequences to the cell. So we have 6 examples here. Transporter proteins are often called channel proteins. They're just tubes. Things go from the outside of the cell in or from the inside of the cell out. They just slip through the tube if the substance can fit. So size is important there. It's got to be small enough to move in or move out of the cell through that protein tube. Channel protein or transporter protein. Same concept. Next to it are the enzymes. Enzymes are proteins. They accelerate cellular reactions. They're very specific with what substances they work with. So it's a shape issue. So that little triangle shape has to fit into the enzyme. The enzyme alters the shape of it, causing a cellular reaction to occur much, much faster than it would normally. We'll revisit enzymes as we get into the membranes and we talk about pathways. Enzymes are essential. If that enzyme shape changes, that reaction slows down, and that can have significant consequences. Cell surface receptors. Little structure there, that blue structure that has a specific shape to it, allows a molecule to attach. The molecule attaches, and it sends a signal into the cell. So it triggers a cellular response. So that those little red dashed lines, that's supposed to indicate an impulse or a response has gone into the cell, causing the cell to do something. So we'll see that with different responses or cell surface receptors. They're specific. They're going to work with only that specific shaped molecule that can attach to it. The cell surface identity marker, this is your ID card. This is unique to you. This is your cell, your identity marker. This is determined by your DNA and your genetics. This is why I cannot donate an organ to a person, because my surface markers are different than yours. And your immune system looks at those surface markers to make sure the cell belongs there. If it finds a cell with a surface marker that it doesn't like, it attacks it. The immune system tries to destroy it. So when people have tissue transplants, often you have to suppress or basically deactivate components of your immune system so that way it doesn't destroy the transplanted tissue. Ideally, you get a tissue that is almost identical to yours with the surface markers, you have less chance of rejection. Okay? If you guys are familiar with autoimmune disorders, your immune system's gone haywire and it's attacking your own cells. It doesn't recognize its own, the body's cell surface markers. To the right of it, cell adhesion or cell-to-cell adhesion proteins. This allows two cells to connect to each other, to attach, to start to create tissues. So same concept we talked about in the previous chapter, previous information. We were looking at cell junctions. Cell-to-cell adhesion proteins help hold cells together. And then the last one are proteins that provide attachment to the cytoskeleton, like anchors, little anchor pegs that allow cytoskeletal structures to attach to it to hold the plasma membrane in place, creating a strong structural component. Okay, so real life example we want to take a quick look at is receptor proteins associated with HIV. HIV is the virus that leads to AIDS. Now, HIV has protein markers on it. Those are those little knobby things there on the HIV molecule. Those attach to your CD4 and your CCR5 co-receptor molecules on your cell membrane, and that's what allows HIV to infect the cell. For some reason, some people do not have the receptor, the CCR5 receptor. So when HIV hits their cell, it can't recognize it, it can't attach to it, and it cannot go into them. They are immune to HIV. So imagine genetically engineering somebody to not have that receptor. They have an immune